Okay, so good news for everybody, or at least good news for me. Our mosses did come in, so we're gonna take a look at some moss today. So I'm gonna orient us to our new group that we're thinking about, and then as you can see on the trays at the end, we are going to fact check whether the Etsy seller knew what they were talking about and compare their product list to the mosses they sent us. That's gonna be our exploratory thing for today, as well as identifying and comparing to some moss I dug out from under the snow on my sidewalk today. So Fun Focus Lab, practical applications to your own lives as well. You can do this with the moss on your sidewalk. Okay, so we're talking about bryophytes today. So bryophytes are going to be our first big group of plants. All right. So as we work through the rest of the class, we're basically going to be walking through a plant phylogeny and talking about different groups of plants as they branch off. So currently, as we've seen in those papers and some of our discussions, there's always some shifting going on, but currently there are 14-ish plants divisions recognized. Varies because people disagree on the algorithms they're using, disagree whether certain things should be grouped together or split apart. Okay, but we are going to be thinking about our land plants. So we got our land plants, and then our first group to branch off here are going to be our bryophytes, which are going to be our mosses, and some liverworts and hornworms. Okay, so this is happening before the evolution of vascular tissue. So before we get xylem and phloem and these vessels that allow us to carry water and nutrients around our little plant bodies. We're going to watch a quick video about our first land plants because it's pretty cool. We can potentially blame them for mass extinctions.
Cambrian period about 5
be dramatic.
years ago, the face of our planet looked completely different. There was land, but there weren't any plants or animals living on it anywhere. Instead, the dry land was rocky and barren with no shrubs or trees or grasses. But clinging to the rocks and the thin ancient soil was life, just a paper-thin film of microbes. These microbes were most likely the only terrestrial life around and had been for several billion years. Scientists think that these ancient microbial films were probably made up of cyanobacteria and maybe some of the first fungi. And each bacterium was likely doing what cyanobacteria do today, sending out tiny filaments of cells from the main bacterial mat to start new colonies. So the fact is, for a good chunk of Earth's history, cyanobacteria had a monopoly on the terrestrial environment. But life on land was about to get a little more crowded, and those newcomers would end up changing the world. Their arrival would make the world colder and fast, and it would drain much of the oxygen out of the world's oceans. Eventually, it would help cause a massive extinction event in which around 85% of animal species, including a quarter of marine animal families, disappeared from the planet forever. This environmental catastrophe is known today as the End-Ordovician extinction event, and it was the first of what we often call the Big Five mass extinctions in the history of our planet. So what could have caused such a massive global calamity? Well, scientists think it may have been kicked off by the world's first tiny terrestrial plant. Now, we don't exactly know what the first terrestrial plant on Earth was, but we have a good idea of what it looked like and how it lived. Unlike animals, plants tend to leave behind a terrible fossil record. You might get a leaf or a stem, but rarely a whole plant. So the earliest fossil record of land plants isn't parts of their bodies, it's their spores, the particles that ancient plants used to reproduce. Pollen didn't exist when plants first made the move onto land, but there were spores, like the ones you see today on a moss or a fern. Back in the 1990s, scientists found lots of plant spores in rocks from Saudi Arabia and the Czech Republic. These spores were dated to 462 million years ago, during that cooling event that took place in the Ordovician period. And they could tell they came from land plants and not aquatic plants, because the spores had a thick covering that all land plant spores have today. This covering protects the spores as they deal with environmental stressors like wind or flowing water. And aquatic plants don't have that because they don't need it in their environment, which tend to be less harsh. And this covering is also what allows spores to fossilize. Long In 2010, even older spores were found in Argentina, and they dated to 470 million years ago. But paleontologists think that the arrival of plants on land actually happened even earlier, based on dates produced by the method known as the molecular clock. By looking at the average number of changes in DNA over time, scientists can calculate when a type of organism evolved on Earth. And this method puts plants on land at least 515 million years ago, right in the middle of the Cambrian period. And it looks like land plants started diversifying almost as soon as they left the ocean. The fossil spores in Argentina weren't just from one kind of plant, but from at least 5 different kinds, a little community of Ordovician plants. It's hard to know what those plants were based on spores, but scientists can tell they were non-vascular, meaning that they didn't have a system of roots and tubes that many modern plants use to move water and nutrients around. Paleobotanists are still debating on what exactly the first type of land plant actually was, but they agree that it was small and moth-like, probably some kind of green algae or liverwort. And these were pioneering little plants, venturing from the water into conditions where they were at risk of drying out. Scientists think that these early plants probably clung to rocks near the water. There they released their spores, taking advantage of the tide to disperse those spores, like their ancestors had done for generations, and gradually transitioning from aquatic to terrestrial life. Over time, through natural selection, they acquired adaptations for life on land, like hard-walled spores and waxy coverings called cuticles that allowed them to become more fully terrestrial. And it looks like their tendency to cling to rocks is what would have spelled disaster for life in the ocean. Today, the scientific name for living material that clings to rocks is cryptogamic cover. And this cover doesn't just sit there, it interacts with the rocks, wearing them down over time and releasing minerals like phosphorus, potassium, and iron. Scientists have used modern cryptogamic covers to see how the first plants might have worn rocks down 500 million years ago. By growing moss on rocks and measuring the minerals released, they found that moss-covered rocks released 60 times more phosphorus than rocks without moss. Once it's freed from the rocks, the phosphorus gets washed away by rainfall, traveling over landscapes and eventually flowing into the ocean. And geologists have found evidence of this very phenomenon in the deep past. In rock formations in modern-day New Mexico and Texas, they found phosphorus in deposits dating to the late Ordovician, when the American Southwest was underwater and just as plants were getting a foothold on the land. And those ancient deposits doomed animal life in the ocean. That's because phosphorus is one of the nutrients that plants need to grow, but it's usually in short supply. Plants can only get it from the breaking down of rocks. So a major influx of phosphorus into the ocean would have caused an explosion of marine plants in the form of huge algal blooms. After algae bloom, they eventually die and are broken down by bacteria, and this process uses up a lot of the oxygen in the water. As a result, the ocean becomes oxygen poor or hypoxic, or even anoxic, where there's no oxygen left. And since marine animals eat oxygen, they can't survive. But that's not the only change that was caused by the phosphorus runoff. A hypoxic ocean can also cool the climate because carbon needs to bind with oxygen to cycle out of the ocean and into the atmosphere as carbon dioxide. But when ocean water is hypoxic, the carbon just gets buried in the sediments and stays there. In the geologic record, buried organic carbon with no oxygen shows up as black shale. And there are extensive black shale deposits in places like China and northern Africa, dating to the late Ordovician. So an oxygen-poor ocean and a cooler climate could have certainly been behind the major extinction of ocean life. Now, in fairness to the plant, experts know that there were other things going on that likely contributed to the extinction event. Namely, it was also a time of massive tectonic activity. New mountains were forming, like the Appalachians, and huge volcanic eruptions took place as tectonic plates of the supercontinent Gondwana moved and folded against each other. Some researchers even suspect that all of the gases spewed out by those volcanoes cooled the earth, causing volcanic mutas. Plus, acid rain likely caused weathering of the new mountains, which removed even more carbon from the atmosphere and drove even more global cooling. But what stands out in the geologic record is how sudden this cold snap was. Starting around 488 million years ago, the planet began to cool, and the temperature continued to drop over the next 44 million years, which is pretty in geologic terms. So something else must have been at work to cause that amount of cooling in such a short time frame. And based on the evidence and modern experimental work, it looks like that trigger might have been plants moving onto land. But there's no need to hate on plants because of all of the downstream effects that came with their big terrestrial transition. Sure, the first land plants were the spark that wreaked havoc on ocean biodiversity, but they also paved the way for all the terrestrial life that came after, because those tiny plants set up the conditions for more sophisticated terrestrial life to evolve. They built up a rich soil base through death and decomposition, and they gradually flooded the atmosphere with oxygen. And over time, the plants themselves took over the land. Their roots became longer to tap deeper for nutrients. Vascular tissues began to carry water and minerals around the plant, supporting the growth of much bigger plants. Later, huge changes, like the evolution of flowering plants, transformed the vegetation on Earth into the ancestors of the plants that we see today and use for food. If it weren't for the pioneering little plants that got a foothold on land 500 million years ago, our planet might still be barren, rocky, and populated by nothing but microbial films. So maybe we can give them a pass for getting the ball rolling on the world's first mass extinction. Do you want more EONS content? Be sure to follow EONS on social media.
We remember to do that,
Definitely.
definitely.
Appearing about 500 million years.
Okay, so today we will be examining the plant villain era, right? Bryophyte. So we're talking about our early non-vascular plants. Really their descendants, right? Because the fact that we can have mosses these days means they have almost certainly been doing something since then, right? They've been evolving continuously. Okay? So when we think about our bryophyte, we think about this movement to land, and we think about why we would do that, right? So if you're an early plant swimming around in the ocean, one of the things you're looking for is light. It gets dark when you go deep down in the ocean, so there's a benefit to coming on land to get that light. There's lots of carbon dioxide, which our plants need as part of their photosynthetic process. Initially, there wasn't anything that ate them on land. Super convenient, fish are very hungry. But we have problems, including a lack of water, and also gravity is a different problem on land than it is underwater, because suddenly you're going to need structures for support if you want to grow up towards the light. A brief note about plants needing water, because this is important for understanding our bryophyte, our non-vascular plants. So water is one of our necessary ingredients for photosynthesis. We have a little cartoon here. At least at the moment, we're not going to go into the deep details of photosynthesis, but we're taking light, we're taking water, right? And we are using them ultimately with some carbon dioxide to produce sugars, oxygen as our byproduct, okay? So we need water for this process, so that's necessary. Another thing that is true is that early plant reproduction requires a moist environment, and that is still true in our mosses, as well as in aquatic plants. So in these groups that we're looking at today, we actually have flagellated sperm from the plant that swim to an egg. So they need to do that through water. So this is some little moss sperm in a petri dish looking around for eggs. Going to look at plants differently. Okay. Brief reminder about mitosis and meiosis. Works the same in plants, kind of, but it's going to be important to remember this as we start to talk about different forms of plants, because we are going to be seeing that some of the things we look at are going to be diploid cells, so 2N. And some of our phases of plant life are going to stick around for a long time in this haploid phase. Okay. So a reminder that meiosis, you start with a diploid parent cell, double pairs of chromosomes, you divide twice, then you get essentially half the genetic information in each of 4 daughter cells. So those are your gametes, and that's indicated by just an N instead of a 2N. So we just have one copy of our genes. So that's our meiosis reminder, because one of the apomorphies, so one of the derived characteristics of land plants, is that they do something called alternation of generations. And we'll look at this more in our fern lab moving forward, but it is characteristic of all plants, right? So yeah, it's one of our really important mutations and adaptation. So when we have alternation of generations in land plants, what we're saying is that plants regularly alternate between two different forms, a diploid form, a 2N form called the sporophyte, and a haploid form, 1N, called the gametophyte. So you actually almost have different plants that you can see, or at least different plant structures as you go through the generations. So that's what this is showing down here with the fern. We'll do ferns next week. It's particularly clear when we look at our ferns. But you'll have one generation as your diploid fern, then you'd have the next generation be a haploid generation, then diploid generation, and so on. So this is a little different than when we think about animals. So the sporophyte generation is kind of in an asexual phase. And then the gametophyte generation, the 1N, is in more of a sexual phase as we are doing plant reproduction. So the difference here is that gametophyte generation in plants can hang around for a long time. Those haploid cells can divide. You can see plants that are haploid. This is important because that is a special characteristic of mosses, is that their haploid form, their gametophyte form, is really the dominant one. So if you're out looking at a moss on a rock, you are likely looking at the gametophyte, or the haploid form of this moss. So we can see the general moss life cycle. For now, we're just focusing on understanding alternation of generation. So we got our gametophyte, the haploid form, and then we got a sporophyte, the diploid form. So this is a cartoon version. This is looking at an actual moss. And here's someone nicely cut and pasted looking at an actual moss under like a petri dish. Now, all of these are kind of paired up. So we're talking about this first radiation off our tree of life. Sometimes these are called the bryophytes as a group. We do want to be careful because currently Bryophyta refers specifically to mosses. So when people use that term, you want to just be a little careful that you understand the context of what they mean there. So we got our hornworts, our liverworts, and our mosses. Today we're going to look at our mosses in lab. But all of these are going to lack vascular tissue, xylem and phloem, and they are a sister group to the rest of the plants we'll talk about throughout this course. Right? So they're branching out first. Right? So everything else would be the sister group over here. So characteristics here are that gametophyte, that haploid generation, is dominant. They still need water for sexual reproduction. So probably think of mosses as wet, and you should, because they kind of need to be to make more moss. Okay. We sometimes say that mosses are poikilohydric and desiccation tolerant. And what that basically means is that because they don't have vascular tissue to carry around water and dissolved sugars, they're just kind of dependent on the water that is in their environment. They can't really control how much water they contain in and of themselves. So that's the poikilohydric bit, that they are just going to have whatever water content the world around them has. And then the desiccation tolerant means that they are adapted so that, like, if they dry out, they don't necessarily die. They can go dormant for a fair amount of time. So they can desiccate and then be rehydrated and spring back to life. So I made a little gif of that happening as well here. Okay. So because they have no vascular tissue, that means they also don't have roots to soak up water and stuff. They're going to need to be small because they do still need water to move around the plant. They just don't have a mechanism of pushing and pulling it. Okay. Right. So when we're looking at a moss, you'll see some little things at the bottom holding them to the surface. These are not true roots. These are structures called rhizoids. Okay. They're often just like one cell thick, very small. Then the main structure you'll see likely is the gametophyte. Okay. So that's the part where you're going to see these little leaf-like looking structures. We'll call them leaves for our purposes. Okay. And if you were to look for the sporophyte generation, the sporophyte is often going to look like a stalk with a capsule on top and then spores coming out of it. But that will be dependent on the species or the group that you're looking at. Okay. a reminder just about this movement of water. So our mosses, our bryophytes, this larger group more generally do still need water. So since they don't have roots, they don't have vascular tissue, they're going to need to get that water through osmosis. So they are literally just soaking up water through their leaves like a sponge. They can also do like a little bit of capillary action. So that's when surface tension between water molecules and the structure they're going up kind of sucks them up just by that surface tension. Okay. So we got 3 groups. We got our mosses, our liverworts, and our hornworts. Okay, so our hornworts look something like this. They're not that common, so don't expect that you'll be likely finding these for your plant collections at the end of the semester. But if you were to see a hornwort, what you would notice is that it has a sporophyte that grows up from kind of a foot. So this flat part would be our gametophyte for the hornwort, and then our sporophyte is growing up in this kind of tower, and this tower would be like filled with spores. A thing that's interesting about the hornworts is that when we were looking at our twigs, we saw that they grew from meristematic tissue at the tip and just grew longer from there. So hornworts are weird in that they do grow from the base, right? That's where their meristem is, which is not how it works in most other plants. Another fun fact about hornworts is that they have cavities that fill up with slime and cyanobacteria that give them nitrogen, which is gross and cool. Fun facts about hornworts. Okay, next we got our liverworts. I love the liverworts. They look like little palm trees, right? So if you're looking at a liverwort, you're likely going to see a big sort of large, flat, rubbery leaf. And then their sporophytes are often going to look like little green flowers, right? Something like that. Now, there are both sallow liverworts. Those are the ones with the large tongue-shaped leaves, and some leafy liverworts. So you will occasionally find a liverwort that's going to look a bit more like a moss, kind of, as we'll see today. Some of our mosses are going to look a little bit like this leafy liverwort here. But the really characteristic, recognizable liverwort that you should be able to tell is not a moss is when you see this big flat tongue structure. So they're our sister group, at least in this phylogeny, to our mosses. Okay. And our true mosses are going to, just in general, look more stem-like, more leaf-like structures. They're going to look more like little trees when we zoom in on them and look closer. So those are the ones that we're going to be looking at mosses today as our lab. So you'll see what I mean, hopefully, as you look at our 8 moss samples and our 9th mystery moss from my sidewalk. So we're going to try and internalize some of the types of structures we see in mosses, some of the variation we see. Our only one division that we want to know for mosses is just to be able to kind of tell, at least for ones that are obvious, whether we're looking at something called an acorcarpus moss versus a pleurocarpus moss. And the difference there is just branches. If you have like a little moss that grows straight up and looks like a tree, you've got an acorcarpus moss. You can see how this just kind of looks like grass, right? That's kind of what an acorcarpus moss would look like. Versus a pleurocarpus moss, so say plural lots, they're going to have lots of branching in a pleurocarpus moss. So these are going to form more like carpet. Okay, cool. And if you're curious, there's a good podcast about bryology with our speaker from Tuesday. She is a bryologist, so she spoke for over an hour on this podcast about mosses, and it was really interesting, but we're not going to listen to that in the lab today because I want us to actually look at our mosses. Okay, so you have a sheet front and back in front of you for our lab today. So we got samples A through G. I started everybody, each table out with a tray of 4 moss samples. So you either have an A, B, C, D tray or an, whoops, E, F, G, H. Sorry guys, add an H, I can't count. To this. You're going to compare that to the list of mosses that this seller said they might send us and see what you think you got, okay? And I want, as you're looking at these, to find an example of something you're pretty sure is a pleurocarpus moss, something you're pretty sure is an acorcarpus moss. It's going to find the most clear example. We don't need to define it for every single one. After you do that, I'll stamp them. You can check what the seller said they gave us. And then on the back, I would like your commentary on whether you think they did a good job or not, whether you have any concerns about their identifications. And then part 3, you're gonna take a look at the moss I pulled out of the sidewalk and let me know what you think about it. Whether it looks like any of these, whether it doesn't, your general thoughts. And that's how we're kind of moving forward, how we're thinking about mosses. And remember that we will be doing collections for the end of the semester. So as you're looking at mosses today, think about what you might look at as we have snow melting and you also pull moss out of your sidewalk, how you might go about identifying that. So on Brightspace, I linked a link to a key of some Minnesota mosses to kind of get you started if you're starting to think about your plant.